Okay, so it's a pleasure to introduce Shalin Naik. Shalin's lab head jointly with molecular medicine and immunology divisions, neither of which I'm in, but I'm the ad hoc introducer. So Shalin's career has been characterized by his interest in understanding how the hematopoietic system is put together and how the different cells relate to each other in terms of lineages and functions and processes. And he's tackled this in a number of different levels. He initially did his stellar PhD with Ken Shortman here at WEHI, where he identified a number of precursors that give rise to the dendritic cell lineages, and these were really seminal papers in this particular field. But that was a predominantly cell biology study, and so Shalin sort of decided for his postdoc to take the next step and to try to understand the hematopoietic system more generally and use a more molecular technique. And he went to Ton Schumacher's lab in Amsterdam and worked on cellular barcoding, and I think he'll show some of that data today. And that again was a very successful outcome for him. And Shalin came back a couple of years ago and last year was appointed a laboratory head. And in his own lab, he's, I think, taken this idea that the relationship between different cell types and the blood cell lineages to the sort of natural endpoint, which is then to try and understand the differentiation events at a single cell level. And he's trying to do this both in a cell biological level by filming and a molecular level by looking at the transcriptome. And I guess we'll see some of that today. So over to you, Shalin.
Thanks, Steve. Is this on? Yeah. So when I was encouraged to give a Wednesday seminar earlier this year, I'd hoped to talk about my work in Holland. In the meantime, I've talked in several different forums, so I'll keep that a little bit short and focus on some of the exciting stuff we've been doing in the last year since we started in the molecular medicine division. So the title of my talk is The Power of One. It's not a play on the power of love, as many people have asked me, perhaps referring to Bryce Courtney's book, but more importantly, that the power of one, or more accurately, many ones, I think can give us novel insights into biology that we can't quite grasp with the population level. So I start off with this slide because we study the immune system. This is a picture of a hand by Cindy Wright, and in the presence of skin, mucosa, and a functioning immune system, this is what we look like. However, this painting by the same artist, entitled Rotten Meat, is what we would look like in the absence of those factors, because indeed we're walking bags of meat at 37 degrees and wet. So let's not forget the importance of why we're studying these cell types. In addition to the generation of the white blood cells, which make up the immune system, and here's a blood smear on there, this is what a normal blood smear looks like with erythrocytes and various leukocyte populations. But if this process of blood cell generation goes awry, of course we can generate leukemias, and many people in this institute work on these diseases. So fundamentally, all of the different immune cell types derive by a process called hematopoiesis, where the current prevailing model, at least the textbook model, is that there is a self-renewing hematopoietic stem cell that differentiates into a multipotent progenitor that then has the capacity to generate every single lineage. And the kind of underlying assumption here is that every cell makes every other cell. Another progenitor I will refer to today is the LMPP, the lymphoid primed multipotent progenitor. And this guy has been claimed to make everything here except the erythroid and megakaryocyte lineage. So I'll be talking mostly about MPPs, LMPPs, and this fraction, depending on the experiment. And we'll be reading out several of these cell types in terms of their development. So the outline of my talk will be to highlight the importance of single-cell studies, talk about the barcoding work from Amsterdam, which is in vivo fate tracking, then basically the results from that made us reflect on what we need to do pushing forward to try and understand those patterns, and start to tackle the evidence for lineage imprinting in MPPs earlier than previously assumed. And then experimental systems to try and identify these imprinting programs. So here's a population of cells that we might be working with, whether it's a cell line or a sorted population. Now, we know that it's made up of a bunch of different cells, and we may put them into various assays that we're interested in, whether it be function, their phenotype, or their transcriptomic signature. So these are just 3 examples, but there are many. In the case of hematopoiesis, that function is actually their fate, what they make. So these are the 3 things I'll talk about today, but you can slot in any aspect of biology into this tree. So when you talk about a population level and its fate, if you transfer a population and it generates red, blue, and green cells, there's actually several possibilities of how this might occur. If you look at the individual cells, you could imagine a situation where every cell made every cell type in equal proportions. Alternatively, there are subsets that are preference towards one or other cell type, or they have a different mix of potentials, and these differ also in their clone size. So these are just 3 examples of an infinite number of possibilities, and I think you'll appreciate that knowing the difference between these can give us novel insights into the biological processes. The other cases of differential expression, when you look at a certain gene by qPCR or a protein level by Western blot and find a 10-fold difference in expression, there are also several possibilities of what might be going on. You might just have every cell expressing 10-fold more in population B than A. There might be a distribution of expression that averages 210, or there might be a single cell that expresses 100-fold more than the other 9, and that can also give you that same result. Now, the challenge in the type of work we're doing is trying to understand what is simply technical noise, what is biological noise, meaning within a certain distribution, and what's a functionally significant change in these expressions and what the consequences are. So our philosophy is that rather than analyze things at the population level, we should analyze the system at the single cell level and then test these for fate, phenotype, and gene expression. Now, of course, flow cytometry has been around for a long time, and so this part has already been established in many labs. Clonal assays, which I'll talk to you about, also have a history, but we've tried to scale these up. And this is a relatively new field of single-cell transcriptomics that we're trying to get up and running. So, there is a history of single-cell hematopoiesis. We're certainly not the first. And the original descriptions of hematopoietic stem and progenitor cells were derived from serial transplantations into mice, where Tillon McCulloch found that colonies represented the progenitor cell or the stem cell that they could transplant or not transplant, depending on the stage. And in that way, they got an appreciation of the pathways of hematopoiesis. Don Metcalf and others developed colony assays in the 60s. And here, every single cell that can generate a colony, you can assess for its particular lineage potential in the presence of different cytokine mixes. A small, but I think valid area worth mentioning is the studies of Ihola Mishka, John Dick, and David Williams, where they actually did retroviral tagging of progenitors, where the integration site of the retrovirus was random and thereby created some sort of a barcode. And these very early studies, where these are populations derived from stem cells, you can basically extrapolate that a band here, if it's shared between 2 cell types, shared a common progenitor. And the relative intensity of that band represents the clonal contribution of that progenitor to that cell type. So this is essentially the precursor of the barcoding I'll tell you about. And Nakauchi of Weissman really pioneered single stem cell transplantations and showed that a single stem cell can reconstitute the entire hematopoietic system. And these have been scaled up both by the Nakauchi labs and the Eves lab in more recent years, doing hundreds of single cell stem cell transplantations, which has revealed lots of interesting stuff about stem cell heterogeneity. So this is a slide back from my PhD with Ken Shortman in 2006, where we were interested in dendritic cell development. So the CDC1, also known as CD8 DCs, you might have heard them. CDC2 are CD11B DCs. We've termed these cells based on a recent opinion piece where we've proposed a unifying nomenclature that crosses tissues and species to try and get rid of this jumble of different names that people have given subtypes. I won't talk about what the function of each of these cell types is today. Suffice to say, they have important roles in different aspects of the immune response. So Ken and I, along with others in the lab, helped to identify a dendritic cell progenitor, which we call pro-DC, otherwise known as CDP, common dendritic cell progenitor, and a pre-DC that could generate all three subtypes. We also developed a culture system whereby if you add FLT3 ligand to total bone marrow and just let it sit for 9 days, you can generate all three DC subtypes. When we took this population of cells at the single cell level and deposited them in filler cells of total bone marrow, and we took these from a green mouse, we could track the fate of individual cells for the first time and ask the question, does every single cell in this population make every three subtypes? Despite every antibody that we tested every antibody in the Shortman fridge, which was quite a lot, and got it down to as homogeneous a population as we could, tested this, and ask the question, what do clones make? So the filler population, so we've got an internal control every time, generated all three subtypes, and I've just color-coded them here, every time at expected and predictable ratios. But the single clone sometimes generated all three subtypes, but only generated one or the other, or combinations of two. And if you put that in a Venn diagram, we find that a very small proportion made everything, and the rest made clones of either one or combinations of two subtypes. So we went back to the earliest progenitor we could think of, where we expected 100 out of 100 cells to generate all three subtypes. And to our surprise, the figure was still only 50%, right? So it came up, but it was still only 50%. And here, these represented large clones, so 9 single cells out of 100 generated a very large clone now, because it's an earlier progenitor, of only plasmacytoids, in conditions that were not limiting to development of the other subtypes. And we know through microscopy that once a single cell divides, it's running around independently of the other, so they're not cross-talking to each other. This kind of gives us an idea that there might be something programmed in there. So my time in Amsterdam was to assess the output of hundreds of single cells, and one could either inject a single cell into hundreds of mice or to inject hundreds of individually marked cells into the one mouse. So we obviously did the latter using the technology that Ton Schumacher had developed in his lab called cellular barcoding. So I won't go into a great detail today about the techniques because I've talked about it previously and it's published. But essentially we have a lentivirus library, each with a different barcode. They're infected into progenitors at a low multiplicity of infection, so there's lots of non-infected progenitors here. And so that it's integrated into the DNA with a different barcode. We then transplant those cells into an irradiated mouse and allow those cells to divide and differentiate as they so desire. At the end of that time period, we then isolate through flow cytometry certain populations of cells, the progeny cells, so cell type A or cell type B, which contains a bunch of cells that derived from one or more of these clones. And then through PCR and sequencing, we can establish which barcodes are found in which cell type, and thereby extrapolate when there were mono-outcome progenitors, bi-outcome progenitors, or mono-outcome progenitor cell type B in this case, as reflected here. So it gives us a semi-quantitative readout of the contribution of each progenitor to each of the different lineages, and we can cross-compare. And on the principle of common versus separate ancestry, we can start to establish what are shared cell types and what are distant cell types. So here's the experimental setup. We sorted LMPPs through the gating strategy for those who are interested. We barcode labeled them in vitro for 6 hours, transplanted them to a partially irradiated mouse, waited 2 weeks, and isolated the 3 DC subtypes, monocytes, neutrophils, B cells, from the spleen, from the bone marrow, monocytes, neutrophils, and B cells. So and then we subjugate these to PCR and sequencing. This is what a hypothetical heat map of the results would look like. So if every LMPP made everything, right, then, as in this case, then you would find a pattern like this. So the way to read this is it's not genes and samples like you might be used to. It's progenitors by row and cell type by column. So if you see a red cell, it tells you that that progenitor generated quite a number of CDC2, but if you see a blue, it didn't generate so many splenic monocytes. And here's the quantitative contribution to the cell type by those progenitors. So it's a really powerful technique to address the output of hundreds or thousands of cells to all the different lineages in a competitive environment. And this is what the data look like, right? So the first thing you notice is there's a lot of black, and that black indicates that not every MPP actually contributed to every cell type. And those MPPs contributed to sometimes 1 or 2 or 3 or more subtypes. And the way the clustering works is it will cluster the cell types according to the progenitor patterns. And in this way, reproducibly, we found that the DC subtypes separated from the classic myeloid and the classic lymphoid lineages, hinting towards dendritic cells representing a separate lineage to these two classic lineages. And when we asked the question, what is your lineage preference for each progenitor, and classified them into bins, we found that very small proportion of LMPPs generated all cell types. It was something like 5%, and their contribution to their cell types was very small. Instead, we found many progenitors that were committed to, or that gave outcome towards the B, myeloid, or DC lineages, or combinations of the two. So actually, one of the challenges we had with this was a custom classifier. One of the challenges is to actually extract meaningful data from this complex data set, this high-dimensional data set, and visualize it. So some more recent work we're doing in collaboration with Jerry Gao and Edmund Crampon is to use different techniques for visualization. I just wanted to highlight this for people who are using these in their analysis, who might be able to use these for their analysis. So this is the same barcode data I showed you in a heat map using TSNE. TSNE stands for, I don't know what, T statistic, something, neighborhood, something. I've got it written down here. I could dictate it, but I won't bother. It's interesting. So Jerry manually gated these populations that he could see by eye, and I don't have time to explain what all of these did, but we found these were uniquely restricted to output towards only one or the other or other DC type, different combinations of two, B cells from either the bone marrow or spleen with a bit of DC development, etc. So this was a really nice way. This is something I'd pictured in my mind's eye for many years, but never knew how to actually extract this heterogeneity because it's non-linear. If you're taking a snapshot of something, you can't assume linearity in that model. So I think this is something we'd like to explore further. So from those studies, what I think we conclude is that LMPP fate in vivo is heterogeneous. And unlike a true multi-outcome progenitor, which was rare, we found the majority of progenitors generated one or two different combinations of cell types and proposed that dendritic cells might represent a separate lineage. However, one can think of different models to explain this heterogeneity. For example, they were all multi-outcome to start with. We transferred in the mouse, they landed in different niches, and that niche dictated what cell type they would produce. Alternatively, it underwent a couple of divisions, and this cell and that one that could make red and green cells died, and you end up with a blue clone. That's another possibility. Or alternatively, there was a program embedded in these progenitors that we are not yet aware of because the tools aren't sufficient or the markers are not known, which dictate generation of blue, green, and red cells. And that leads me to the hypothesis of our lab and the work I'll show you today, which is that fate is imprinted much earlier in development than is commonly assumed through novel molecular programs. And we do not yet know what these programs are. So the aims are to establish the systems in order to test these hypotheses, which is quite a large challenge in itself. And if correct, if the hypothesis is correct, identify what those programs are and to validate them through CRISPR, shRNA, or small molecule inhibitors. So I won't talk about aim 3 because that's basically the stage we're up to now, but I'll talk about aims 1 and 2. The significance of this is, well, it gives us an explanation of how diverse tissues are generated. I'm talking about the blood cell system, but this could equally apply to any heterogeneous cell tissue that you're concerned with. And how early are programs actually becoming active is an open question. Is this just stochastic heterogeneity or functionally relevant heterogeneity? That's important to know. Is how deep do we subset? If you think about lymphocytes, for example, Mitchell and Miller first separated them into B and T, and then others separated them into CD4 and CD8, and Tregs and T helpers, right? So we've gone historically down the field and know what all the different subsets are. But could we have known about them a priori right at the start without any prior knowledge? And finally, some of these factors may be applicable to tissue manipulation therapeutically to either wind down or wind up cell numbers that are relevant to a disease or biology. And there's no evidence for this yet, but we've thought, and we'd like to test, and open to anyone who might be able to help us with this, could these programs that are as yet identified actually be the etiology, because it's so early in development, for certain leukemias and lymphomas? And perhaps some of the factors that we identify are the ones already established in leukemia models. It'll be interesting to cross-compare those. So what is our test for imprinting? We came up with this simple idea. If we drop in a single cell, and rather than waiting till its end clone, we split it into two separate wells or two separate mice and ask if they generate the same cell type, in this case red cells. Or it could be the generation of green cells in both replicate wells, blue, but if it generated a discordant outcome in the two separate areas, we would hypothesize that this was not an imprinted fate. So essentially what we're asking is, do twins do the same thing? If we were to separate twins and put them in a different environment and ask them a question, was the answer to a question or the action that we test them on already imprinted in them, or would it depend on their environment? And obviously it's going to be a combination of both. So how do we design that experiment? So what we did was take LMPPs, barcode label them, and then culture them for a period of 3 days so that there were multiple sisters derived from the single cell. This culture period did not affect the distribution of the barcodes in the different cell types through control experiments, and there wasn't a bias in one fate or the other, so we knew the system worked. Then we randomly assigned the daughters, basically mixed it up in the well, and injected it intrasplenically into two separate mice to localize their development, because we're dealing with small numbers. And after 2 weeks, isolated total dendritic cells, myeloid cells, or B cells from one mouse or the other. If there was imprinting, we would expect the same barcode to end up in the same cell type in two mice, because it was inherited by the daughters. Whereas if environment plays a greater role, that might be random, which barcode ends up making which cell type. So when we analyzed this experiment, we found that it was a little bit, as maybe expected, a combination of both. But what we could do for the first time was describe which progenitors were imprinted for which subtype. So here you see an example of a lot of progenitors that generated dendritic cells in both mice, but did not generate B cells or myeloid cells. Here are a bunch of progenitors that generated B cells, but not other cells, and some that generated DCs and myeloid cells, but not B cells. However, there are some instances where progenitors had a discordant fate. And when we statistically tested whether this was greater than expected by chance by randomizing the barcodes in one mouse, we indeed found that there was a high proportion that were conserved in their lineage bias across two mice. So this is the summary I showed you before. Now we can say, based on the clone splitting experience, that there's actually some, as represented by wedges here, some programs that are dictating these fates. Now, what is the nature of these programs? So we run into a Heisenbergian problem. So for those who watch Breaking Bad, this is Heisenberg. He didn't just pull the name out of a hat. It refers to a chemist named Werner Heisenberg, whose Heisenberg uncertainty principle is actually often misconstrued as the observer effect, which I'm going to misconstrue it again. But the principle states you can't both measure an object without affecting the nature of that object. And so we face a problem, a Heisenbergian problem, is that how can you measure the output of a single cell, for example, if it made red cells, and go back in time and find out what made that cell special to make those red cells? Perhaps those programs have already been lost from the progeny cells, so you'll never know what they were. By contrast, you could send a single cell for RNA-seq or some other measure, but then you've lost it to measure fate. So again, through the clone splitting principle, we've come up with this approach, which is reverse hematology. So we drop in a single cell, we grow it up as a small clone, and then split it into 3. 2 of these go for fate assessment in conditions which will allow you to detect different fates, and 1 set of cells we save for RNA sequencing. We then do that not just for 1 cell, but for dozens and hopefully hundreds of cells in the future, and then find out, after culture in this particular case, which one made red, which one made blue, and which one made green cells, and then look for the genes that correlate with red cell fate, blue cell fate, or green cell fate. All right, so now you're linking fate with gene expression, which is something we weren't able to do prior. So in the case of DC fate, these are the experiments we did, and we dropped it in, split them in, and looked for fate at 2.5, looked for the fate at day 8 for these DC subtypes. So here's just an example of one little clone. We dropped in a single cell, we let it grow 2.5 days, then we just pipette up and down, split it into 2 wells, take an image so we can quantitate the number of cells, let them grow for a further 5 days, image them again, and then do FACS analysis. So we've got the numbers at every step. We can monitor a lot of different things. So here's some examples of the clones that come out of that assay. So the filler cells, remember, in every case of a single cell, there's always bone marrow filler cells that always make roughly equal proportions of all the cell types, or of all the DC subtypes in this case. And these are the GFP negative. But the GFP positive, we can identify or classify them according to their bias, clones that made CDC1, CDC2, plasmacytoids, or all 3. So in this way, what we looked for a plot which would allow us to tabulate that heterogeneity and came up with the idea of a ternary plot, where for any clone that can add up to 100%, we can plot it as such. So if this clone generated 40, 40, and 20% of these 3 subtypes, we can draw off the axis here, 40% for PDCs, 40% for CDC1, and 20% for CDC2, and draw a point. So that single point represents the clonal output of that single cell. So this is actually the point. So this is analysis done by Peter Diakomis and Melanie Barlow. And what they did was take the population average, put on the plot, and we first asked the question, if we were to randomly draw cells from this proportion relative to the experimental values of clone output, where would those clones sit? And this is what we found. As expected, they kind of center around the average. However, when we looked at the real data, it looked more like this. So the experimental data showed quite a few clones, and Peter's jittered them here to fit into that axis here and here. But many of the progenitors are actually quite towards the apices, apexes, whatever. And there were some that generated all 3 in the middle. But these all represent clones that had a sister. So we can draw a line between the sisters and ask, is the distance very short between the clones, or is it very long? Now, Peter one thing to note is that all of these did add up to overlap with the population average, so that the clones aren't behaving in some spurious way. If you add up all the clones, they behave as the population. Here are some random sister assignments, and as you can see, it's a big spider's web of fates. But if we now link them to their relevant sister, we get rid of a lot of the spider's web, but there are some cases where the lines are long. But a lot of the cases of short distances you just can't see because it's harder to see shorter lines than longer lines on this plot, and many here, and here are hard to see. But we can do statistics on these. So Peter did a one-sided 2-sample t-test looking at the Euclidean distance between sisters and non-sisters and found a highly significant correlation, highly significant short Euclidean distance, which is indicative that DC subtype preference is largely imprinted. That is, if you split them into 2 wells, you get these sisters, which then sets us up to say, now we have a system that allows us to do RNA sequencing on some of the sisters and find out which genes correlate to that. So single-cell sequencing is a protocol that we've been working on for a year to optimize it to our purposes, and we finally got it working. I won't go over all of the technical details or the quality controls, and it's actually a work in progress still. Suffice to say, we sort single cells using a fax machine into a 384 well plate, which contains 1 microliter of index primer. We sort it directly into that. We lyse it at 70 degrees to release the mRNA, polyadenylated mRNA. In that well, each cell has a poly-DT primer, but in addition to that, there's a little 8 base pair index for which we can know which well has which index. So that when you generate the cDNA from this process, you have the index attached to every transcript from the yellow, red, etc. Because they're all indexed, we can pull them after the cDNA step and process them as a single sample, which is great because it's technically very easy. And through aRNA amplification, in vitro transcription to amplify the RNA linearly, fragmentation to get it to the right size, adapter ligation to prepare it for the library, and the PCR, we can then send it for sequencing. So in our proof of principle experiments, Jessica Yaring with bioinformatics analysis done by Matt Ritchie, we then dropped in 15 single cells, 10 lots of 10 cells, 5 lots of 50 cells, and 2 lots of 100 cells, and asked the question, can we get reads from every single cell we deposit? And is the read count proportional to the numbers? And indeed, we could. This was a particularly shallow read count, but we're now doing a deeper run to extract all of the data. And if we had wells with 0, we had a couple of wells with 0, we find a couple of reads, but these reads are presumably due to sequencing errors of the index that bring them down to that read count. So then we applied the RNA sequencing to the siblings in our reverse hematology assay for DC fate. And this analysis was done by Toby Sargent with the genes selected by Matt Ritchie. So Matt Ritchie looked for genes using, let me read this, linear regression models to identify gene expression that correlate with fate with an FDR of less than 0.05. And then we basically went through those genes, and just for today, I'm going to show you an example. We got a list of 160 genes that all look interesting, but we're still confirming them and validating them. But just as an example, I'll show you a couple. So we have our sisters from our experiment. We can connect them in space based on their sister relationships, and then we just average that for the sake of representation. Then overlaid on top of that, Toby measures gene expression correlated with that fate. So here is the expression level, both in terms of a dot size increase and color intensity. So here's one gene, for example, named TCF4, which in the siblings didn't look to be differentially correlated between these clones. And this is kind of surprising because TCF4 is a gene implicated in plasmacytoid development, also known as E22, and we didn't find any great correlation of fate there. Another example is IRF8, which we know to be important for plasmacytoid and DC development, and there might be a slight correlation here, and in fact, we need more data points, but we didn't see anything obvious. It didn't come out in the screen. What did come out in the screen were some other genes. One example is BRD3, and this was correlated with plasmacytoid fate. BRD3 is a bromodomain protein that's a histone, acetylated histone reader. And another gene that came out is JADE1, that's histone acetylase writer. So JADE1 is a partner of HBO1, and very little is known about any of these two molecules in DC fate, let alone biological processes in general. So we're now interested in confirming whether these hits are true and validating those. Another example is MS4A6C, which is a cell surface marker that correlated with conventional DC fate but was not on progenitors destined to become plasmacytoids. So these are all genes in the earliest sisters that are correlated with fate. Whether they're causative for fate or predictive of fate is something we need to test. So we can do this for the DC lineage, but we can actually do this for as many cell types as we care to test, as long as we have enough sisters. So Jessica, again, did these experiments with the help of Yaring, where she dropped in a single progenitor and pre-expanded them with KIT ligand, FLT3 ligand, and OP9s, and then split those daughters now not into 2 wells, but into many different wells. And these wells drive the development of either B-cell differentiation, T-cell differentiation. We gave some of the cells to Don to test in colony assays. Now, because the colony by definition is like a well, so we don't need to split them in 2, right? As many colonies you have is as many wells as you've tested. And DC fate, and we will in the future send these for RNA-seq. So here's what the results look like. This is a population level control where 100 progenitors were grown up for 3.5 days, then split into all those different assays. And as you can see, the population could generate plenty of B cells, plenty of T cells, the different myeloid colonies, and dendritic cells. But then when we looked for the output of single cells, we found, for example, this is just one example, from a single cell and its daughter split, they could generate a few B cells and B cells assays, but lots of T cells, a few blast colonies, but not so many myeloid colonies, and no DCs. In another example, we have a clone that generated B cells in both technical replicates, not so many T cells and no myeloid cells and DCs. Here's a clone that generated dendritic cells only and no other lineage, and a clone that generated myeloid colonies, but no BT or DC. And here's the closest we could come, actually, to a multi-outcome clone. We didn't see any that generated everything. And this is the closest we could come to where it generates BT, lots of macrophage colonies, but no other ones, and dendritic cells. So we did this not just for the clones here, but we've done it for 192 clones over 2 experiments in a stellar effort by Jessica, who just came in 1 day a week. She also sorted out the single-cell sequencing. So she's been an amazing asset to the lab. I want to say that right now, and the acknowledgements as well. If you see her, please encourage her to do honours, because she's thinking of taking a year out and chilling out, which is fine. But so this is how we've plotted the lineage bias, like the heat map I showed you with barcoding, but now we're looking at single cells in vitro, and now we can talk about potential. In vivo, we talked about outcome because we don't know where they land, but here they're put in assays that unequivocally drive B-cell development, and we did not get any B-cells. The caveat to that, hence why there's an asterisk, is that there is a clonal proliferation. So very early on, there could be commitment happening before clonal expansion, but it'd have to happen prior to the first division, at the first division, but not much after that. Otherwise, the sisters would not show the same fate across different wells. So this is what the plot looks like, and so you read this by saying there's a bunch of progenitor cells that made B cells but nothing else. These guys made T cells but nothing else, etc. And the way it's clustering is also putting the DC subtypes together and the myeloid cells. But it's not as clean, maybe, and we probably don't have enough data points yet. But what Toby, I'll get to that in a sec, but if we can ask the question, how many cell types does each clone produce? So that's the percentage of progenitors here, and the number of cell types produced by a single progenitor. And as you can see, none made 12, none made 11, 1 made 10, and then the numbers start to go up. That is, the majority of progenitors, when split in this way, only generated a few different cell types and not everything. Of course, this is a progenitor population we know is heterogeneous. We started off with that. We didn't want to make any assumptions of markers, but this is the first systematic analysis of what we found. And amongst the 192, we actually see nothing making 11 or 12 cell types. This is a distance measurements between the sisters that Toby did, and as you can see, sisters have a shorter distance, therefore are more related in their fate than non-sisters. And this has a high p-value, low p-value. And this is a technique that Toby's using to try and disentangle the heterogeneity there as well. It's like TSNE, it's called local linear embedding. And in this way, Toby observed that there was like a T-shape to the trees. And when he overlaid each of the cell types on top, we could visually see that these were clustering plasmacytoids. These progenitors represented ones that were myeloid bias, and these would be in T bias. So to formally show that, he just overlaid the colors of each of the cell types. And as you can see, these tend to be more red, these tend to be more green, the myeloid colonies, the blue of the megs here. And these tend to be more greeny, yellow, orange, which represent the B and T. So again, through a different assay, not barcoding, so there's no caveats of the lentiviral transduction or irradiation, we're still observing the segregation of myeloid, DC, and lymphocyte fate, which we think is quite interesting and will pursue. All right, that leads me to my summary. I showed you that many progenitors are heterogeneous in their fate, and that due to the clone splitting experiments, we know that this involves some degree of programming. I've also shown you systems that now we've established in the last year with some preliminary data that allow us to identify what those genes are involved in that lineage imprinting process. Select here. Now we want to go ahead and knock out or knock down the various genes and look for an absence of red cells, blue cells, and green cells to hopefully figure out what the molecular programs are that dictate all of these cell types. And another thing that we'll hopefully discover is that when you do a population of cells and look for its transcriptomic signature, you see a gene network. You don't know whether that network was present in every cell or that it represented, for example, 4 populations with 4 different networks. And this type of analysis will allow us to disentangle those possibilities. So this is my last slide, and the take-home message, if you remember nothing else, that I hope one considers that studying things on the single cell level, whether it be for function, phenotype, or gene expression, and you can lob in anything here, whether it's epigenomics or phosphorylation status, whatever, can be very valuable in understanding biology. And most importantly, a clone splitting approach doesn't just tell you what those values are, it actually links each of them to each other so that you can look for your favorite genes, find out what fate they had or phenotype they had, and try and disentangle that heterogeneity. So the last slide is the acknowledgement slide. So this is the lab as it stands. Nick is finished honours last year, but we included him for the photo. So Yaring is the kind of champion of lab. He kind of takes care of everything to get the systems up and running and helping the students and postdocs to set up and doing many, many experiments of his own I haven't talked about. Nick did all of the DC clone splitting stuff along with Melanie and Peter to establish that fate bias is intrinsic. Dawn is an honours student from this year who has decided to continue to do a PhD, so hopefully you'll hear from the stuff she's doing in 4 years' time. Jessica, as I said, is a very talented Europe student, so remember that face and go harass her, who single-handedly got single-cell sequencing up and running, the clone splitting experiments, fax analysis, etc. Daniela, we've recently recruited. She's a German heritage Israeli, and she's come from a next-gen sequencing background. So we're hoping to get our transcriptomes from, currently it's about $20 to $30 per cell transcriptome. Our target is to get it to $1 a transcriptome so that we can do the single-cell analysis in a high-throughput manner. But please don't come knocking on our door just yet. We still need a few months to scale it up and validate it. Sangeeth is a recent PhD trainee in the lab, and Gabrielle's a recent Europe student as well. I'd like to acknowledge Ton Schumacher and Layla for all of the barcoding studies that I showed you today. I returned to Phil Hodgkin's lab when I first came back, and actually Phil co-supervised all of the work on the clone splitting work of Nick and all of the Hodgkin lab members. We have joint meetings, and they're an absolute asset to have for us for them to critique our work and have discussions. Thanks to Don and Ladina for the colony assays. I highlighted where the absolutely superb work of Toby and Matt has been in actually trying to extract this complex single-cell data. Melanie and Peter have done a great effort in identifying that statistical significance. Doug for his support in the molecular division, which we are. Jerry and Edmund for this visualization and other work that we're doing. Steve, for the sequencing facility, has really helped us get that single cell up and running, single cell seq. Simon and the other members of the FACS lab, and Merle Dayton for animal work. These are the funding sources that I had both during my postdoc and currently. And thanks for your attention. And the other divisions which I'm joined with. Thanks.
If you cluster on the single cell expression data, do you get the same clusters out of it as clustered on the barcoding or the clone splitting?
We haven't done that, but we've talked to others who have, and they say that the transcriptomic data is too high dimensional to actually capture that. They've given it a go, but I'm very happy to give it a test. Excellent. Sorry, Edmund's nodding, saying we can definitely do that, so let's do that.
Shalin, is there a pluripotent stem cell? And if there is, you need to demonstrate resistance in your assays for them to have validity.
Yeah. Yes, so the population control works, so the assays themselves are fine, but what you're saying is, ideally you have a clone that can generate everything so that you know the assay works at the single cell level. Do we need to find one? If it doesn't exist, I would argue no. How do we... right? There's a possibility, well, we know from single cell transplantation that you can get a single cell regenerating all lineages, so it does exist. This particular assay is not capturing stem cells because the stem cells don't proliferate very much in that day, so actually we're biasing our assay towards progenitors at that stage. We have in barcoding work, I have, but I won't show you. It's in the paper. We do find stem cells that make everything. So from the barcoding aspect, we have demonstrated that a single cell can generate all lineages, and in fact, we find that stem cells, there's often a rare stem cell in that population, and it generates lots of everything.
The concept of lineage imprinting is interesting in a system that needs to be as dynamic as hematopoiesis. What do you think happens in emergency hematopoiesis? Is it a matter of those few multi-outcome clones dominating? Does it overcome lineage imprinting?
So that's a really interesting question, and what Dawn is pursuing in her honours project, which you can hear about, she's talking next week in the student seminar series, so you can have a look at what she's found there. And we've just, I think, started to address these issues. And you can imagine three possibilities. One is that the same progenitors that were already there in the steady state just make more of something, so that's increased clonal expansion. The second possibility is that there's recruitment of progenitors to the process, right? So that would tangibly result in an increased number of barcodes. And whether that were from dormant progenitors or progenitors that could be from dormant progenitors or those that would have made something else. So if they were on their way to making something else and you hit them with GCSF, for example, do they shift their fate? And that would be a case of cytokine-induced lineage divergence. We can tackle those questions with barcoding, and that's, in fact, what we're doing.
Shalin, you're looking for some imprinting program that predetermines this. Could it possibly be the other way around, that there is a program that helps maintain multipotency, and that if you lose the program, multipotency is then an unstable situation and you tip one way or the other, and it happens already by the time you look at those daughter cells. Is that a possibility?
Yeah, it is a possibility. I hadn't actually considered that, and I will, and what that data might look like. So you'd presume that... So we'd look not just for genes that are correlated with fate, I guess is the answer, but also inversely correlated with fate, and that might get to that question, I think, but I have to think about that.
Just because a particular stem cell doesn't produce cells of every possible lineage, of course, doesn't mean that it couldn't have potentially. Have you considered, I mean, there might be a stochastic process of lineage splitting, and it just might be very unlikely to produce, it might have the potential to produce everything, but just stochastically it might not happen very often. Have you, so one question to answer is, if every stem cell did have complete capacity, how many would you have to observe before you saw one producing every possible outcome?
So that's an important question. There's two ways we're tackling that issue. One is that we hope through our screens to identify surface markers that will allow us to prospectively isolate lineage bias stem and progenitor cells. So by sorting for a population that unequivocally has no potential for something else, I think that's demonstrated that at least for those cells, they can't make anything. Your second question is whether it's a stochastic reason. All I can say is that we can test, in addition to the clone splitting, we'll also be doing single cell deposition pure from a fresh bone marrow sample. And there, if the cell lands in conditions which allow for the potential to be seen, and if we see no potential for that cell, it might have died, that's true. But what we can do is use index sorting, which is a procedure where you know the phenotype of every cell that went into every well, and label them with as many markers as possible to try and hone down where those cells sit. So without a priori knowledge of their fate, we can find out what they are. Still doesn't answer your question, and I think we haven't thought of a good way to unequivocally say that cell could have had potential for everything. We can't unequivocally say that yet.
There are signaling pathways that require neighbors, such as the notch signaling pathway, efferent signaling. Of course, when you make signal cells, you disrupt them, and when you provide them with their own autonomy as neighbors, may or may not establish, if something like that was required, you may or may not establish it quickly enough in the rather unlikely case where you then get potency or not.
Yes.
So I think the single cell steps.
Yeah. So the caveat we always state is that at least prior to major clonal expansion. So what that basically implies is prior to that cell's isolation from the bone marrow, prior to its first division, perhaps at the first couple of divisions where we could stochastically lose a fate, and I draw that example of cells that die, then you might have two sisters interacting with each other physically and dictating that fate. However, after that division, and we validated this with microscopy, they are separate. And they are in conditions, the filler cells, for which they're contacting multiple niches and running around independently and dividing. So at least after that stage, they are independent, they're contacting multiple niches, yet the program remains. So
60%.
I can't remember which data you're referring to, but yes, it's not a complete, there's a proportion of clones that are biased and a proportion that are not. Yes, indeed. So we don't argue that all cells are all imprinted, it's just proportion are, and I won't go into the model today, but our model suggests that branching doesn't just happen at fixed time points or fixed progenitor populations, it can happen throughout the whole hematopoietic process. And when we capture a snapshot of a population and see the heterogeneity, we've seen things that did split earlier and things that are about to split soon after.
Would you get the same thing if you transplanted into non-irradiated mice?
So we've tried those experiments using the c-kit hypermorph mice, where they have a competitive advantage from Warren, the WWV mice. And we haven't had good engraftment in order to study those small numbers. I think you still need to inject 1 million cells or so to get some detectable progeny. We might retest it again with larger numbers, but now we're getting to cell numbers that are much larger than our barcode library. So we could either change libraries or whatever. What we're separately doing is trying to generate barcodes inside the cell, inside the mouse, using some fancy Crelox-P machinery, where randomly you get flipping that generates a barcode in the DNA of the cell inside the mouse. So no isolation, no transplantation. And in fact, there was a Nature paper that kind of scooped us on that a couple of weeks ago from Fernando Camargo's lab, where they used transposon jumping as a barcode and found that, well, there's a few caveats to their study, that there were many progenitors contributing to the cell types at a given time, and that was a very dynamic process. And they extrapolated that the stem cells are actually doing not much in the steady state. Progenitors do most of the work, the stem cells are just sitting there, the kind of spare tire model. where they don't do anything until danger is called.
All right, last one.
Could the culture condition modify the outcome of the experiment?
Indeed it does. It definitely modifies the outcome of the experiment towards the lineage we're testing for. However, because we're testing for all possibilities, if it can't make B cells in B cell conditions, then it can't make B cells, unless you obviously experimentally put in PAX5 or RAG machine or something. So yes, it does, but that's kind of the point, I guess.
Okay, let's thank Shalin.